This is a recent EMBO journal paper from the Leung lab on PARP14-mediated ADP ribosylation in p62 bodies under interferon stimulation. I find the paper interesting, and I want to take a deeper look at the changing genes and relationships than what's covered in the manuscript itself. The data is public, so let's pull the raw files and reanalyze them through our own lens. The dataset is on Proteome Exchange under PDX 057944. 12 DIA runs, A549 cells, p62 immunoprecipitation with and without interferon gamma. That's all I need. Switching to Europa XP, quick login, and we're in. All data enters through the Files tab. Beyond direct upload and S3, there's an external sources option that pulls from Geo and Proteome Exchange directly. Server-to-server transfer, no local download needed. Beyond pasting an accession, Europa XP also offers AI-enhanced search, where you describe the experiment in plain language and the system finds matching datasets across Proteome Exchange. Today, I already have the accession, so I'll paste it. The summary you see here is the one Proteome Exchange ships with the dataset. During retrieval, our AI also generates an augmented summary that captures sample groupings and design details, often beyond what's in the structured metadata. We'll see that augmented summary do real work when we get to annotation. I want the raw files so I can run my own search. I'll filter to raw, select all 12 samples, and click Upload Files. The transfer happens in the background, so I can close this dialog and keep working. The files are in. On arrival, every mass spec file is automatically pre-processed and a 2D image is generated as a quick visual QC. In tile view, I can scan all 12 at once. The signal looks consistent across runs, no obvious dropouts, and no truncated gradients. We are good to go. Now that we have all 12 raw files uploaded, we can create samples. First, I'll add the accession as a sample prefix, and I'll toggle Create Study after sample creation. There are 3 modes available: existing library, library free, and Carafe enhanced. For this analysis, we'll use Carafe enhanced for better detection. Next, we select the FASTA file and the digestion and modifications. Note that the AI parameter assistance has already pre-filled most of this based on the manuscript summary, so I'll just verify that it's correct. Interim files produced by DIA-NN are saved at the sample level. You can access them from here. You can also review the signal after processing using the heat map. Now that our 12 samples are processed and the study is created, let's open it. Before proceeding with the analysis, let's first review the summary about this dataset that was prepared for us by an AI assistant. We are using additional data from the manuscript and from the internet if necessary to get information that is not found in the repository. The first tab shows the dataset overview. The next one shows the sample metadata, which will come in handy in a second when we are annotating samples. Next, we have sample preparation, then the mass spec parameters used, and then details on data processing from the manuscript. Finally, you can see key findings from the study. We can now open Annotation Manager and apply sample annotations to this study. We can use the CSV file we were just looking at, match files, and merge all the annotations from the file. That's the metadata step done in about 10 seconds instead of minutes or hours. We can now easily create groups using loaded annotations. For now, we'll only compare 2 groups, p62 untreated and p62 interferon gamma. The IgG control is useful later for quality control and specificity assessment, but that's not the subject of this video. Default view is the per sample box plot where distributions look comparable. We can check all by all correlation as a fast sanity check. The Pearson's correlation is above 0.9 within group, with an expected drop between treatment groups. There don't appear to be any technical outliers. Before normalizing, we will subset to the 8 p62 samples. The IgG controls have much lower overall protein load, so including them in the per sample median would skew normalization for the p62 runs. Filter by selection creates a new brick called p62 only. We can select this brick and then filter on expression and require at least 50% of samples and at least one group to have valid signal. Next, we select the filtered set and we'll median normalize this subset. Finally, we'll use MinProbe for imputation of the missing values, which in DIA sit below the detection limit. To visualize the data, we can run PCA on the normalized data. We can see here there's clear separation along PC1 between untreated and interferon-treated samples. Now we're ready for differential abundance testing. We can run a t-test on the two p62 groups with Benjamini-Hochberg p-value correction. Since the upstream data is linear, I set the transformation to log 2, so the test takes the log internally. We can also perform gene set enrichment analysis in the same step. While we're here, let's do another t-test on the data before imputation so we can compare results before and after MinProbe. We can view both t-test results as volcano plots. We can see many significantly changing peptides. We can use the slider selection tool to filter the results. We can click points in the volcano plot to view a box plot of the peptide abundance. Top hits include PARP14 itself, the central player in the paper. Switching to Europa XP Free Volcano Viewer, peptides higher in the interferon-treated group are in red. Since we did the comparison at the peptide level, we can get much better insight, and for each protein that changes, we can examine all of the peptides and how they behave. Clicking on one peptide selects all peptides from that protein. We can spend a lot of time here putting a microscope to a number of genes of interest. Next, we can review the GSEA analysis. Opening the pathway dot plot, we can find interferon gamma response pathway and select it. The volcano plot shows the location of peptides from proteins that map to that pathway. We can see that they are all increased in interferon treated, as expected. Once again, there's a lot to explore. Single protein view opened from the MinProbe dataset gives us access to every protein and every peptide. Search PARP14. The protein sequence is divided into 50 regions, each colored by peptide intensity in that region across all files. We can group samples by condition and easily see that peptides in PARP14 are higher in interferon-treated samples than in the untreated group. We can zoom in to the peptide we are interested in and click on it to see it highlighted on the left. Selecting the View XIC button gives you the extracted ion chromatogram for MS1 and the top 6 MS2 peaks with a lot of additional information. If you're satisfied with the assignment, you can mark this peptide as verified. We can use this later to filter peptides. We can also open single protein view from the t-test results instead of normalized data. The view inherits the selection, so only significant peptides are visible. This is a way to quickly go through and validate results. Well, there you have it. That's a complete reanalysis of a published DIA proteomics dataset, raw files pulled from Proteome Exchange, processed through DIA-NN, normalized, statistically tested, pathway analyzed, and validated at the peptide level. Every protein and every signature available for inspection. Total hands-on time, less than 2 hours. This is just a brief overview of what you can do in the Europa XP OmicsCloud. There is so much more that is available. Call us today to set up a demo or get your subscription started.